Alright, if you've made it to this point, you should have already completed your Week 5 assignments. Last week, during NTI Week 5, we learned about aseptic technique and bacterial isolation. I demonstrated the process of streaking bacteria onto an agar plate with disposable inoculating loops, and you also watched a video that demonstrated aseptic technique and its application in the lab. Now we are going to focus on our very last new topic of the semester before we learn how Anna died. Bacterial morphology. The term morphology looks and sounds more complex than its meaning. Morphology is the study of form. Essentially, bacterial morphology is the study or observance of bacteria's physical characteristics. Here is an agar plate that has been streaked with bacteria. As you can see, the streak marks are faint but visible. However, there are no visible bacteria. After we streak the bacteria, we have to give them a chance to grow. At this point, they are still individual microscopic bacterial cells. But under the right conditions, the bacteria will grow and form colonies or circular groups of many bacterial cells. Bacterial colonies are visible to the naked eye. Bacteria utilize the nutrients in the agar for metabolic processes, which lead to growth. We incubate the bacteria to increase the rate of growth. Bacteria will grow at room temperature, but we typically see accelerated growth when bacteria are incubated at body temperature, which is approximately 37 degrees Celsius. This is not true of all bacteria, but will be the rule of thumb for bacteria that we grow in MI and BI in the years to come. After the agar plates have been incubated for 1 to 2 days, we would remove them from the incubator and observe the growth. As you can see, there are many variations of bacterial growth. This is because there are many different species of bacteria. If you look closely, you'll notice that most of these plates have the same growth pattern. The areas where bacterial growth is dense correspond with the first streak. The individual colonies or circles appear where the third and fourth streaks were made. In these images, we see yellow bacteria, white bacteria, red bacteria, bioluminescent bacteria that glow in UV light, and the bottom middle image shows white bacteria on a red agar. Like I mentioned in the previous presentations, there are many different types of agar we could use. We typically observe 4 different physical characteristics: color, margin, elevation, and the size of the colonies. Bacteria come in many different colors: white, red, pink, yellow, cream, translucent. Each species of bacteria is a specific color. For example, E. coli is an off-white or beige color. However, there are many off-white beige bacteria. This is why we have to observe characteristics other than color to identify a particular species. However, color is a great starting point. Another physical characteristic that is included under bacterial morphology is margins. This is the shape of the edge of the colony. There are many different margins. For the purposes of this course, we classify margins as circular or wavy/irregular, rather than try to say words like rhizoid or filamentous. 2 examples are shown. The yellow bacteria on top appears to have circular margins, while the white bacteria on the bottom has wavy/irregular margins. A third characteristic we observe is the elevation of the bacterial colonies. Elevation describes the side view of the colony. This is probably the hardest thing to observe, especially in pictures. The 3 bacteria pictured are all different elevations. The furthest left is flat, or maybe even slightly raised. The bottom image, or the bottom bacteria, are convex, and the top right is umbonate. Lastly, we observe the size of the colonies. You can do this by simply comparing 2 species. Species A is larger than species B, etc. But we also measure the colonies like you see in the image. Depending on the species, some colonies are classified as large, while others are classified as medium or even small. The size is constant. For example, most E. coli colonies are classified as small. Here is the bacteria from Anna's urine. Observe the color, margins, and elevation. The bottom right image is the most magnified. In the chart, you will see several different species of bacteria and their morphological characteristics. Use your observations of the bacteria from Anna's urine and the chart to determine which species of bacteria was present in Anna's urine. Make your final determination on the Google form that is linked to this assignment. If you don't correctly identify the bacteria on your first try, you may try again, or as many times as it takes. The Google form submission will conclude your work for this week. Email me with any questions.